Hello everyone, I am Aswathy K, advised by Dr. Karthik Raman of Department of Biotechnology, IIT Madras. Today I'll be presenting our work on constraint-based method to identify function-specific minimal microbiomes from large microbial communities. Microbial species exist in large communities and exhibit biological functionalities. Oftentimes, all the species present in the community do not contribute to a given functionality. It's often possible to identify a smaller microbial community, which is a subset of the large community, that's capable of the functionality that we're interested in. Such smaller communities are called minimal microbiomes. Minimal microbiomes need not be unique. It's often possible to identify multiple minimal microbiomes for a given specific functionality because some species in the large community would have functional redundancy in spite of being dissimilar in nature. Identifying such minimal microbiomes are useful for designing treatment strategies for diseases caused by disruption of gut microbiome. It's also useful for bioengineering applications. So we have developed a systematic mathematical method for identifying minimal microbiomes from large microbial community capable of certain functionality. By functionality, we mean production of certain metabolites, say certain SCFAs. Our method is top-down and constraint-based approach. This is a formulation of our method. In step 1, we find the maximum growth rate of the large community using joint flux balance analysis, which is a linear programming problem. In the second step, we find the maximum metabolic production for the growth rate obtained in step 1. This is a flux variability analysis, and it's also a linear programming problem. From these steps, we obtain the constraints required to identify the minimal microbiome. In step 3, we systematically delete organisms from the community. We remove an organism and check for the growth rate and metabolite production constraints. If they are satisfied, then we delete the organism. If not, we keep it and move on. In the fourth step, we minimize the L1 norm of membership vector of the resultant smaller community. This is a mixed integer linear programming problem. The size of this MILP will depend on the size after the third step, after the deletion steps. The third step is optional. It not only helps reduce the size of MILP, it also helps, it's also needed to identify multiple distinct minimal microbiomes because the identified microbiome will depend on the sequence in which deletion was done. The MILP formulation is as follows. The objective is to minimize the L1 norm of the membership vector. The components of the membership vector are 0 or 1. 0 indicates that the member is not present in the community, and 1 indicates that the member is present in the community, subject to the stoichiometric constraints and bounds on the flux values. Fluxes are the continuous variables, and the membership components of the membership vector are 0 or 1 binary integers. These are the constraints specified by the large community. The exchange flux of an SCFA has to be greater than or equal to fraction of SCFA exchange flux in the large community. The biomass flux in the minimal microbiome has to be greater than or equal to a fraction of the biomass flux from the large community. Here is a simulation result of a 25-member original community on high fiber diet for butyrate production on these parameters. We were able to identify 80 minimal microbiomes, and here is a plot of the frequency of occurrence of the species of the large community in the minimal microbiomes. We know that the bacteroid species are capable of digesting dietary fiber. That explains why bacteroid species are occurring at a high frequency in the minimal microbiomes. Also, because of the functional redundancy of certain species, we see that some of the species are occurring at a lower frequency because they are replaceable. To conclude, large communities can be reduced to function-specific minimal microbiomes. We are presenting a constraint-based method to identify non-unique minimal microbiomes. We're calling it a flexible algorithm because a lot of the parameters can be input by the user. Presence of species capable of digesting dietary fibers is observed in the minimal microbiomes. Identified minimal microbiomes can be mutually exclusive. The algorithm can quantitatively predict metabolic fluxes. It's reasonable for a coexisting community because of the constraint-based approach used. The gut microbiome is known to be a coexisting community, so it's reasonable for that. The minimal microbiome code is available on our GitHub page. I would like to acknowledge Professor Karthik Raman for guiding me, IBSE for the fellowship, the postdoctoral fellowship, and the travel grant, and all the members of Raman Lab for the suggestions and inputs. Thank you.